Hello friends, welcome to Biology EXtrema. We will teach biology from the basics to the extreme. Myself Keshav Bansal, and today we are going to study about triacylglycerols and waxes. Okay, so let's start. So first we are going to talk about triacylglycerols. These are what? Both of these triacylglycerols and waxes are lipids. Okay, if we talk about them, these are lipids. And what are lipids? What are lipids? Lipids are, if we define lipids, these are those substances which are insoluble in polar solvent, okay, like water, while there are solvent in polar solvents like benzene. So there are soluble in non-polar solvents like benzene. Okay, that's one way to define these stuff, because they don't have a common chemical structure. Clear? But still, if we want to define the through structure, we can say that these are esters of fatty acids and alcohol. So what do you mean by that? When we join a acid and alcohol, we get ester. Okay, and here if we join a alcohol with the fatty acid, okay, then we get an ester. Okay, so there are two components in this structure. There is fatty acid and there is alcohol. Clear? Now we have studied it, we have already talked about it in our previous videos, that the lipids, they can be classified as simple lipids and complex lipids. In simple lipids, what we have alcohol attached to these fatty acids. Okay, and if we add some additional groups, like phosphate, okay, like some oligosaccharides, then these simple lipids become complex lipids due to addition of some additional groups. Okay, so like today's video, we have to study triacylglycerols. Okay, we can call them triacylglycerols, triglycerides. These are the same thing, triacylglycerol, triglycerides, or we can just call them tags. Okay, tags. So these are simplest lipids. Okay, these are in simple lipid category, tags and waxes. Okay, and even in simple lipids, there are simplest lipids. Okay, tags. So they just consist of alcohol and fatty acid. There are no additional groups present. Okay, in additional groups, they can be, for example, phospholipids. Okay, they can be sphingolipids. So these are all other additional groups like glycolipids and complex lipids. So today's topic is tags and waxes. Okay, so as I told you, we have two components, fatty acids and glycerol molecule. So if we talk specifically about triacylglycerols, we have one glycerol molecule. Okay, through one glycerol molecule, let's say this is a glycerol molecule. Okay, we have three fatty acids attached. Okay, we have these three fatty acids attached to it. Okay, this one alcohol. Okay, glycerol is an alcohol. Clear? So this is a polar structure, while these fatty acids, these are non-polar, because as we know that lipids are amphipathic molecules, means they have both polar domain and a non-polar domain. Okay, so polar domain is this alcohol part, which is glycerol in this case, in the case of tags, and the non-polar part is fatty acids. Okay, and now you know that these fatty acids are large group, they are more bulky in comparison to this glycerol. So that's why we sometimes say that this is non-polar. Okay, but we always remember that lipids are amphipathic molecule, they have both the domains, polar and non-polar. Okay, so due to these larger non-polar domain, they will be hydrophobic, because the non-polar part is hydrophobic. Okay, and the bonds, because we know that lipids are esters, so these bonds through which fatty acids are attached to glycerol, they form ester linkage. Okay, they form ester linkage. If somewhere it is asked which kind of linkage it is ester. Okay, now we divide these tags into two types. Either these fatty acids, these three fatty acids. Okay, either they can be same, they can be of one type, single type, or they can be of two or more than 2 types. Okay, they means they can be of different types. As a result, the tags can be divided into simple and mixed tags. Okay, now in comparison to water, they have, if you talk about their physical properties, they have low specific gravities. They are called neutral lipids. Why they are called neutral lipids? Because they have no net charge. Okay, they have no charge actually, so they are called neutral lipids. Clear? Now we define, we categorize, again classify or categorize triacylglycerols into 2, fats and oils. How we categorize them? Let's talk about it. We know that these consist of glycerol and fatty acids. So what we do, we will focus on fatty acid. Okay, as we know that fatty acids can be saturated or they can be unsaturated. What do I mean by that? That saturated fatty acids, they have single bonds. Okay, they have single bonds in the hydrocarbon chain. Okay, single bonds. While in unsaturated fatty acids, they have double bonds. Okay, what happens because of that? If we take, let's take this saturated fatty acids. These saturated fatty acids have single bond in them, and as a result, they are compactly packed. They are more compact. Okay, they are more crystal-like structure. So what happens? That high energy is required to break them, to destabilize their structure, and as a result, melting point will be higher. Okay, as a result, melting point will be higher. But in the case of unsaturated fatty acids, due to the double bond, we have a kink in the fatty acid. In the hydrocarbon tail, we have a kink like this. This is the kink, and as a result, these fatty acids, when they are present, they are not packed properly. Okay, they aren't packed properly, and as a result, they are not much compact. So less energy is required. Less energy is required to destabilize them. Okay, so as a result, the melting point will be lower. So what we get to know from all this discussion, that saturated fatty acids have high melting point, while unsaturated fatty acids have low melting point. Okay, and as a result, if you take room temperature, so at room temperature, because it's not that much high temperature. Okay, so at room temperature, unsaturated fatty acids will be in liquid form. Okay, like you have seen oils, vegetable oil. Okay, and in case of saturated fatty acids, they will be solid, like ghee or tristearine or beef fat. Okay, so these saturated fatty acids are called fats. Okay, these tags which are rich in saturated fatty acids are called fats, and these are solid at room temperature, while the tags which are rich in unsaturated fatty acids, and are liquid at room temperature, are called oils. Okay, so now we know the difference between fats and oils. Okay, also these tags are used as energy reserves. Okay, these are mainly used as energy reserves in the lower organism. In a lower eukaryotic cell, they are just present in the cytosol as oil droplets, while in vertebrates, in higher organism, there are specialized cells dedicated to store these fats, which are called adipocytes. In plants also, in the seeds of plants, they are present as oils. Okay, so as energy reserves. Clear? Now, when these lipid rich foods are exposed to oxygen, because oxygen has oxidative power, so what happens, that they oxidize the double bonds present in unsaturated fatty acids, and as a result, these lipid rich foods get spoiled. Their taste and smell get spoiled. Okay, their taste becomes unpleasant. Clear? This phenomenon is called rancidity. Okay, so exposure to oxygen results in oxidative cleavage of double bonds of unsaturated fatty acids in the lipid rich food, and this phenomenon is called rancidity. The food is now spoiled. It has become rancid. Clear? Now if we talk about partial hydrogenation, what happens, because we know that these oils, okay, these are going to use in frying and the other stuff where they are going to be exposed to high temperatures. So we need to make sure that they are more stable at that high temperature, okay, because they have low melting point, they are liquid in even in low temperature, so they need to be stable at high temperature. So what we do, because they have unsaturated fatty acids, okay, and even in unsaturated fatty acids they are mostly composed of cis type of double bonds instead of trans fats, okay. So what we do, because in biological system cis type of double bonds are more present, they are more abundant because they are more fluid in nature, okay. So cis double bonds, we convert these cis double bonds into single double bonds, means from unsaturated fatty acid we can form saturated fatty acids. This is partial hydrogenation, okay. But sometimes what happens, due to partial hydrogenation, these unsaturated fatty acids, this specific, if we say specifically, these cis double bonds are converted to trans double bonds, okay. In cis double bonds we have a similar group on the same side, while in trans we have a similar group on the opposite side, okay. And trans fatty acids, they have lower fluidity, okay, they are more rigid, so they, I mean they have less, they have higher melting point in comparison to cis double bonds, okay. So we know that saturated fatty acid, if we compare saturated and unsaturated fatty acids, saturated fatty acids have higher melting point, okay. But if we take unsaturated fatty acids, they can be cis and trans. So trans fatty acids have higher melting point in comparison to cis fatty acids, okay. So but these trans fatty acids, when they are this undesirable effect that they are formed during this partial hydrogenation, so what happens, they are, because they are associated with cardiovascular disease, because they increase the level of triacylglycerols in the body, and lower density lipoproteins, means bad cholesterol, while they lower the level of high density lipoproteins, which is good cholesterol, okay. They also cause inflammatory response in the body, okay. So ultimately they are harmful to organism, clear. So and you can find these trans fatty acids in like in fast foods, okay, the deep fried stuff. So it's better to avoid them in your diet, okay. Now I will talk about saponification, okay. What happens if we take triacylglycerols, these tags, and we add some alkalis to it, okay. We add some alkalis like sodium hydroxide, potassium hydroxide. What happens, these alkalis, they hydrolyze, they break down these tags into fatty salts of fatty acids plus glycerol, okay. These alkalis, they break down tags, they hydrolyze it into salts and glycerol, okay. This process is called saponification, and the milligram of these alkalis required to hydrolyze 1 gram of tags, I am repeating, the milligrams of alkalis required to hydrolyze the 1 gram of tags is called saponification number, okay. So this is it. Now if we talk about iodine number, so how it is used to find the degree of unsaturation, okay. The number of iodine you can add to a 100-gram sample of tag, okay, any tag, it can be fat, it can be oil, okay. So if we add if we take the how much grams of iodine we can add to fat and oils, okay. Let's say let's say for example we are taking hypothetical numbers, we are able to add 50 grams of fats, 50 grams of iodine to fats, okay, to a sample one, okay, which can be fat, we don't know that now yet, okay. And to a sample second we are able to add 80 grams of iodine number, okay. So what happens, that the more gram of iodine we are able to add in a in our sample, okay, in our sample of tag, the more saturated it is. So if I give you the choice which one of these two, sample 1 or 2 is fat or which one of these is oil, you can now determine using these iodine number, okay, that which is more unsaturated fatty acid, because the more iodine number we can add, the grams of iodine we can add in the sample, the more unsaturated it is. And we know that oil is more unsaturated and fats are made up of saturated fatty acid, so they will we can just add a small amount of or lesser amount of iodine in the fats. Okay, so this is the way we'll tell the degree of unsaturation. I hope you get it. Now we'll talk about waxes. So waxes, in waxes they are just similar, like they are simple lipids as in case of tags. Okay, they're just little difference that here we use high molecular weight monohydroxy alcohols, means there's only one OH group present in the alcohol. Okay, they cannot be more than one. And while triacylglycerols are used mostly as energy reserves, there are multiple functions of waxes. Okay, they are used as energy stores, they are used as water repellents. Like in the case of birds, they have purine glands, so what they do, they put they rub their beak over these glands, and then they lubricate their feather, they make them waterproof. Okay, and even if you talk about tropical plants like holly, poison ivy, yeah, okay, so they also have these waxes which they are using to prevent evaporation, to protect them against parasites. Clear? Because they prevent their entry, the entry of parasites. And also we use in other industries like pharmaceutical, cosmetic. Okay, there's bee wax. This is from animal, so their source can be animal and their source can also be plant. Okay, like carnauba wax. So this is about waxes. And keep in mind that these waxes, their melting point is higher in comparison to triacylglycerols. Okay, so this is it. I hope you like the video. If you like the video, please share it, subscribe our channel, and thank you from Biologia EXtrema. See you next time. Take care.